No, I was tapping the wrong thing.
AC is okay.
Okay, we'll start, and this is, as I just mentioned to you, it is completely unprepared, but the reason I want to give this talk is because in our lab, this is not what we normally do, okay, the kind of simulation that I'm going to talk about, but yet at some point of time when in around a few years ago, we had some experimental data. And we thought it would be useful to, do a computer simulation. And, I thought about it a little bit, and, I talked to students in my lab, and they were kind of hesitant to do it. But then, even in a lab where, completely experimental lab, we actually could do something which we thought was quite nice within a short period of time, using a master student in my lab, and Arvind, who did it. So I therefore thought I would share this with you. And let me give a little bit of background because I've not talked about this. So you've heard about that motors generate force and this and that. So we wanted to simulate a situation which is something like this. Okay, and we knew from various experimental data, which I will not go into right now, that on single endosomes, okay? All of you know what endosomes are, I suppose. Even if you don't know, these are things carried by motors inside cells, okay? On single endosomes, and just to give you an idea of the scale, this size can vary, but think of it roughly as 1 micron. Okay? 1 micron. So we'll roughly start with that size. On single endosomes, which were moving either inside cells or moving when we took them out of the cell and put them on a microtubule, we could see that there are, because my drawing is bad, I will just write K and D. Okay, you could see that by other experiments, you could show that there are kinesin and typing motors attached to these endosomes, and the resulting motion from the activity of both of these motors, we could measure that actually. And so what we found is that if you make a trajectory, let's say again this is time, okay, and this is, let's say this is a microtubule, okay, and let's say this is your minus direction and this is your plus direction. You know it by some it doesn't matter how you know it, okay. What we found that these endosomes would move over long distances in this direction, and they would, something would happen when they would reverse. We'll come to that in a bit. And then they would move over shorter distances in the plus direction, and you know, it would go on. Sometimes they would move. This is the kind of motion that we saw. Okay, that there was a net bias in the minus direction moving towards this, but there were instances where it would go for short distances in the other direction. And what we found is that in these segments where something would reverse direction, that was the interesting part, because why? And so what you are seeing here is obviously bidirectional motion, right? Sometimes it goes this way, sometimes it goes that way. And the question is, what gives rise to this bidirectional motion? Okay, and there has been a lot of controversy in the field. And when we looked at this, we focused on what is happening at these reversals, because that is where things are changing, right? So when we looked at this, if I zoom in into this, I saw, I'll draw this. We saw something like this. So this is zoomed in here. Okay. So yes. Okay. So the length scale here, think of this as, I'll draw a scale bar here. Think of this as 3 microns or something like that. Okay. So this reversal, so this is length. Okay. So this would be about, so this is about maybe 4 microns, 3 microns. This would be shorter. So when we zoomed in here, and so how is this data collected? I just really want to give you a flavor of how to correlate experiments with some theoretical work. So this data is collected by looking at a movie of this thing moving. Okay, you have seen Ambarish. Where is Ambarish? Okay, there he is. He showed you a movie of something moving along a microtubule, right? So you track those things, and you find out the position at time intervals. Okay, by tracking that, and just so that you remember this, we tracked this object, the endosome, at a frame rate of 30 frames per second. Everybody understand what frames per second means? You found its position 30 times in a second. Therefore, the time between two dots is how much? 1/30th of a second. Okay, so or 30 milliseconds roughly. So now when we zoomed in here, we had data like this, and what we found is that in this segment, which we called a tug of war segment, I will call this TOW, the velocity was smaller, which you can see because the velocity is the slope and it is smaller here. And secondly, these endosomes, which were spherical here, okay, they would stretch in this direction. You could see them stretching, okay. By seeing this, what we inferred from this was that these motors, so single endosome on this had both kinds of motors attached to it, and then they were actually physically pulling against each other, and that's what we inferred. Now, interestingly, when we looked at this segment, so see, this, you can either look at something here or you can look at something here, right? So this is a M and this is P. What is M and P? Minus plus. This is a minus to plus reversal. Okay. Similarly, you can have a plus to minus reversal. This one. Is everybody understanding this? Okay. Now, irrespective of whether it was a minus to plus or a plus to minus, the slope of this region, I should maybe, I'm just exaggerating it to be a little more, but this region was always, almost always negative. Okay. Now, this negative was not an actual velocity. What was happening, and I want you to appreciate this, is that we were tracking the position of this object, right? And when you do a video tracking, you find something called the centroid of this. Where is the center of this mass? Okay. Now, when this thing stretches here, that your program will find some center of mass, because now it is not a sphere anymore, right? It is stretched. Now, what was happening in these regions is that this endosome was held fixed at one point, but stretching in the minus direction. You understand that? So there were teams of, these were the kinesin motors and these were the dynein motors, let us say, okay. What was happening is that when this stretching was happening, the kinesin motors held it at one point, and this end, so this end remained stationary, but this end elongated. So this position did not change. This end elongated. And therefore, the center of position shifted from here in this direction. And therefore, the velocity seen was, the apparent velocity was negative. Is this clear or not? It was an artifact of the way you track the position of an object. It was not a real velocity. The object was stationary, but its shape was changing. Therefore, you saw something like a velocity. Because the track position was at the center of this, and if you hold it at one end and elongate it at the other end, you will find the center here, and if you elongate it more, the center will be here like this. Right?
And size of this retina will be such independent that it will presume that there is some change in the velocity of it or direction of this retina.
No, I did not understand your question.
But I think to say this size, L size is very less of it.
1 micron, no, yes.
So, if size thing happens, then there is change in the mass of that particular body.
There is no change in mass. There is change in shape.
Change in shape.
Yes.
Leading to the somewhat illusion that you are in shape is partial opposite level. Would that be significant that?
No. See, we are talking about this region now. Okay, and again, when it goes in this direction, it is again spherical. So only in this region there is a pull by both kinds of motors, and there is a shape change. After it has gone beyond this, what has happened here is that when you are going like this, there is only one kind of motors who is pulling the thing. Okay, these are the D motors because it's going minus. One at this point of time, some K's attach. Attach where? To the microtubule. They were already on the endosome. They attach to the microtubule, and now suddenly the D's field, somebody is pulling me back. So the whole thing kind of slows down and stops here, and that is where you see this deformation, and that's what I talked to you about. What happened here was the D's detached. Okay, and now it started moving in the opposite direction because of the K's. Am I answering your question? If not, maybe I can talk to you later. Okay, so is everybody understanding this? Okay. No, okay.
So here when minus 2 plus reversal.
Minus 2 plus reversal, yes.
There is a negative slope reversal.
Right.
So is it, plus 2 minus there is a positive slope? Is it?
Now let me complete what I was trying. Maybe I will not complete. So what this was telling us, so that the slope was negative whether you do minus to plus or plus to minus, told us that in this region the minus motors were winning this tug of war. but the plus motors would not let go. Okay, that the plus motors, let's say if we measure them, they just stuck somewhere like this, and the minus motors are tenaciously pulling this like this, and so therefore this thing is stretching, and therefore the center of mass is going from here to here to if it is like this, and therefore you see a negative slope, and that is the same whether you go like this or like that. Okay, but these are the details of this. That's not the main point of my talk. This is what we inform. So what we wanted to do is that looking at all this, one of the things was that there was a, so now what is the question that we were trying to ask is that, look, here is a situation where you have a net bias in motion. What is that bias? It is in the minus direction. Yes.
So, the velocity, the negative velocity which we are seeing here is only due to the change in the center of mass, or due to the movement also. Both is held, and it is also.
So right. So we actually track the end positions of these objects also. Okay, and this position, okay, changes really very little. So it is, there may be some contribution from actual movement, but it is largely because of deformation. Okay, so there may be a 5% contribution because of actual movement, largely because of deformation. Okay.
So whether it is the particle is moving in antrograde or retrograde, the slope you saw was always increasing.
Yes, that's what I, yes.
So even in the retrograde, it's
right. So whether it was minus to plus or plus to minus, the slope was always negative.
But suppose the particle is moving in angular direction, means Carnation motors are taking it forward.
Like this situation.
Then doesn't it happen that dynein motor now will come and then try to pull it from the back, means, and then Carnation, we should see the centroid moving towards the.
No, that's what I'm saying. So what was happening here is that the team of dyneins was winning, and therefore, irrespective of whether you did this or that, the team of dyneins was winning, and therefore this position of the centroid preferentially moved towards the minus direction.
One, what is the aspect ratio change between the sphere and the
25%? 25% change from the original size. 25%. I'll just once again I can check and tell you, but from what I remember, it was 25%.
When it changes direction, the sort of intermediate protein, the thing is coming back almost instantaneously. It's retracting back into the sphere almost instantaneously.
That's an interesting question. So you're talking about, so depending on whether it's a minus to plus or plus to minus, I have to look at it. We saw a recoil in one of these situations. Okay, and I have it noted down, but it's been a while. I can get back to you on this. So depending on whether you go this way, that way, in one of the cases you see a recoil, and it makes sense. The direction in which you see a recoil, you actually saw that. Yes, that's a good point. Okay, what was I trying to say? Yes, so you saw a minus bias in the motion here. Okay, now what we wanted to understand is that, so you had these situations where you had a tug of wars. Okay, this. Now occasionally during these situations, you would see this guy breaking into two. Okay, and you can think of it as fission of endosomes, which people have observed. People know about quite well. Okay, and this, once they break up, these would either go that way, or something else would happen. But they would actually break apart, which also shows that, look, there was a mechanical stretching force. Now, what we wanted to understand here was, how do you achieve this kind of a balance? Okay, how do you achieve a net transport in this direction, which is important because endosomes have to move towards the center of the cell on an average to fuse with lysosomes so that things are degraded. Okay, so biologically you require this motion towards the minus direction. You don't want the whole thing to move towards the plus direction, except in exceptional cases. Okay, so you want one net minus. Okay. 2, you want these tug of wars to happen occasionally. And the purpose of this tug of war is to sometimes break this into two. Okay, fission. All right. Why do you want to do that? Because by breaking endosomes into two, what people think happens is that you can recycle endocytic receptors. Let's not get into that. Okay. There is a biological reason for this thing to break into two, and so you want fission, but you don't want this fission to happen all the time. Okay, because so in other words, if you had the motors always pulling against each other, you don't want that situation either. Because A, then you would get nowhere. B, then everything would be breaking apart all the time. You don't want that. Okay. So how do you get a situation where you bias the motion in one direction and at the same time have occasional situations when you enter such a tug of war and you have this kind of fission events. Okay. You don't want to overdo either. So for this, what we proposed was that the tug of war, so this, what I'm showing you here, corresponds to this state. Okay, just remember that. But what we said is that what is happening actually is not this. Okay, what is happening is something different, and one could, you people know how to make a Punnett square. Okay, one could make something like a Punnett square and put in possible values of kinesin and dynein force, and what would be the consequence of that. And what we realized was that only one arrangement allows both of these to happen simultaneously. And that's what we wanted to model and try and understand, and that's what I want to talk about if I have the time. Now, to get both of these two together, what we realized was required that instead of those guys pulling like that, you need one kinesin who is one or two kinesin who is strong, opposed by, and this is supposed to be a row, okay, by multiple, roughly 5 or 6 dyneins who are weak. Okay. Now this arrangement gives you this. Okay. And we wanted to show that in a model, because does it make sense? Because we have been hearing for a long time that if you have multiple motors, your processivity increases, right? So if you have multiple dyneins, let us say, you will go on an average in the minus direction, and fewer kinds will go on average in minus direction. Everybody agrees with that. So net minus we are able to do. Okay. Fission. Now for fission, why will fission happen? Because when things pull against each other, right? So for fission, there should be a meaningful tug of war. Now, for a meaningful tug of war, the opposing teams should be matched in force, and not only in force, in tenacity as well. How long can you sustain a force? Right? Just applying force is not enough. How long do you sustain that force is also important. So these two teams should be matched in tenacity and force. For that, if I have 5 or 6 weak dynein motors exerting about 1 piconewton force each dynein motor, and this guy exerts 5 piconewton, okay, then I reach a force balance roughly, right? Okay, so I am now, if I reach a force balance, then I may be able to have this fission kind of a thing, and I'm also doing this. Okay, let's think of alternative scenarios. Yes.
But in that graph, kinesin is not changing the position.
Kinesis is not changing the position.
The upper end is stays there.
See, frankly, we don't know exactly where on the endosome the kinesin or the dynein is. Nobody can.
Whether a kinesin is acting the same dynein is action.
We believe that. That is something which given the optical resolution, nobody can, really.
Consider those conditions that kinesin is a person and dynein.
Sure, you can do that. Under that situation, what can also happen that the whole endosome can rotate. Because remember that these things are moving on people which have multiple parallel protofilaments. We hypothesize that happens, but we have not directly observed it. But it's possible because it's a multiple lane highway, remember. Yes.
The presence of this endosome give similar, the two are not differentiated. The two endosomes cannot be differentiated.
Right.
Can be differentiated?
No, not right. Differentiate on the basis of what?
No, if you're seeing it and you're measuring the distance with respect to the time, then once this divide endosomes, one is going in a positive direction,
Right,
and one is going in negative
Right.
direction.
Right.
So if you cannot differentiate the two endosomes, so
So what we can differentiate, let me tell you. So when they go in opposite directions, we don't see that happening all the time, let me be very frank. But when we do see it, the ones which were going in the minus direction move over longer distances. The ones which go in the plus direction move over shorter distances and detach, which is what you would expect if the ones which were going in the minus direction had more motors on them, which is what I'm telling you. Right? Okay?
Do we have evidence that
Okay, I'll never get to the modeling part. Yeah.
Regarding the fusion process.
Right.
So, where do you put the fusion machinery into the motor pulling?
No, you have two teams. You have two teams which are pulling, and the endosome has certain mechanical properties. Maybe, and you pull and the rope will break somewhere. That's what I'm trying to say. You take a rope. These two teams are pulling.
So that I understand the pulling
Right.
thing. So
Right.
if I remember correctly, endosome fusion also depends. I mean, there are machinery which regulate the fusion of endosomes.
Yes.
So where do we put that? Because if you download those machinery, endosome fusion is affected. So does that?
So those are higher order regulatory mechanisms. Those mechanisms about fission, I don't know precisely what you're talking about, but there could be higher order regulatory mechanisms which regulate the activity and the number of these motors. Okay, then you would regulate the fission. Do you have any specific protein or any specific process in mind?
I had a discussion with Alok and we
Right,
and
right.
So the idea was, so suppose if I assume this is similar thing in case of peroxisomes or mitochondria, they also fuse and fission.
Sure.
But if you like protein like DRP, they are required to break peroxisomes, right? So if the DRP recruits dynein,
No, DRP may, for example, change the membrane properties. Okay, it may, like dynamic. Okay, it may, make a pulse-like thing like this, and
Yeah. So here in this case we don't have. So if we don't require those proteins here.
we don't think we require it because all this is, we can reconstitute all this in vitro. Okay, where we have not added these motor proteins. So what we believe is that this is a purely motor-driven process. Now the actual dynamics of this, how this will happen, may be changed in different situations inside cells. Certainly.
Surprisingly, nothing comes out of the lumen when you break two cells, just like that.
It is, if something maybe we can't see it, but roughly if you look at the volume of the two chota ones, it is similar to the volume of the parent. Roughly, we haven't really gone back and looked at this. Yeah. But all this is happening on a microtubule in vitro, so there is nothing else. No, so that you understand. Okay. Okay, so anyway, so once we did all this, we hypothesized that for this whole thing to work, you would need multiple weak motors of one kind. I will call them W motor, this dynein weak, okay, and kinesin strong. Multiple weak motors in opposition to a single or a few, very few strong motors. Okay. Now we wanted to see. Okay, so wait, hold on. No, so not just multiple weak, and that's not all. I also mentioned the word tenacity, if you people remember. Okay, so the sum of these tenacities should roughly be equal to the tenacity of this one, not just the force. How long can each of, let's say 5 are in opposition to 1. So a single dynein should be able to sustain whatever force it generates for 1/5 of the time of kinesin. Okay, roughly there are catches, tricks to this, but roughly you can understand like this, that the sum total tenacity and force of this should match the force and tenacity of this one. Okay, so we hypothesize that these guys should have a lower tenacity. the time for which they can sustain force should be less than this guy, and they should be weaker than this guy, then all this business will happen. All this will happen, okay? That is what we wanted to model and see whether that happens. Now, when we did this, what we wanted is that we wanted to take, so what is a Monte Carlo model? I mean, you have heard about that, and you basically assign probabilities to things, okay? And you assign probabilities and you run your computer and you start at some time and you watch after whatever 10 minutes time, and there are multiple pathways to things. Let us say, okay, and maybe I have, yeah. So using a Monte Carlo simulation. So this is the paper that Amrish was mentioning, that you can make a decision based on many probabilities. Amrish showed you one where there were PA, PB, and nothing happening. So you can have many more probabilities. Okay. So it can, it's used widely used in particle physics. You can use it in branched enzymatic pathways because there are many possibilities of an enzyme, as it goes through its cycle. And if you, say that this pathway has this probability and that pathway that probability, you can model this whole process. Okay. So what's the problem? Okay. So what we wanted to do then was that we wanted to use this kind of Monte Carlo simulations, which you are sufficiently educated about now, to understand how, given the properties of this motor, you could simulate a situation like this. Okay. So now the problem is that what do we start with? You need experimental data. Okay. So what is the experimental data you need to model this? And maybe you can add what maybe I have it written somewhere. Okay, yeah, good. So so think of the program as a black box. Okay, it's not black; it has many colors here. But you think of the program as a box where you give it some inputs and it will give you some outputs. Okay. So what are the inputs that are needed to understand this? So you are going to. So what should be the output of my program? Is the question. The output of my program, I want it to be this trajectory. Okay, let us understand this first. And this trajectory should include situations where things are not pulling against each other, situations where things are pulling against each other, all of this. And I want this to run for whatever 10 minutes or so and see what happens at the end. Okay. So I will need to simulate situations where nobody is pulling against anybody. Here. So when when this is going in the minus direction, okay. So what do I need to know now here? I need to know the velocity of dyning motors, right? So I need to know the velocity of dyning motors. So if I know that, let's say 2 microns per second, I will start here now. I need to know if I start here, what is the likelihood of a minus run ending? How long do I go on? When do I stop? Okay. When will I stop? Either I will detach and fall off. That's one probability. The probability for that is low. Let us ignore that probability right now. The other probability is when it attaches, right? Then something will change. I cannot move freely anymore. Okay. So I now need to know, to end this, what do I need to know now? Tell me. Attachment rate of kinesin, right? I will have to find that from somewhere, and how we'll find that, we'll get to that in a minute. Now you enter this state. I'm just zooming in here. Now when you enter this state, okay, you can, to model how much it stretches, you can use the elastic constant of endosome, but let's not worry about that right now, okay. Our concern is, okay, now we enter this state. When do I get out of this state? It has to be the detachment, because I'm going to go plus now. I need the detachment rate of dynein, not single dynein. Whatever was the number of dyneins, of those number of dyneins. Okay? And now I go here. Now I will move. I know there is no dynein now. I go like this and I need the velocity of kinesin. Which yes.
Is it certain that when the endosome is under the tug of war condition, it will reverse its direction?
No. Good point. So, so there are many situations. Okay, I have not plotted it properly. Okay, so there are many situations. That's a good point. There are many situations when you see a tug of war and you keep going in that direction. Okay, I should have mentioned that if this happens. And the probability of reversal versus continuing, we think is roughly 50-50. Okay, we did not see a strong pattern. So there were times when you get stuck, and then but you after some time you continue with the same direction. But there are times also where this could have happened. Okay, and that was roughly 50-50. That is a little difficult to estimate experimentally for reasons which maybe we can discuss later. Okay. Yes, just a summary. Yes.
So attachment and detachment here is the motor to kinesin, motor to microtubule.
To microtubule. Yes. So so attachment and detachment rate means here is the cargo, here is the motor. The motor is not detaching from the cargo. The motor is detaching or attaching to the microtubule. Okay, just be clear about this. Oops. Yeah. Okay. Yes.
Whether there is any increase in number of motors which are already attached to the cargo whenever there is.
We don't think so. We don't think so because what we did is that during these segments, when they were moving over long distances uninterrupted, we stalled it at many places and measured roughly the same force. Do you get the? Do you understand why we did that? Because that means that the motor number is not fluctuating. There will be some variation, but it's not changing hugely.
Not even at the point where there is tug of war during this.
There we can't measure the stall force, so we don't know. But if it doesn't change here, no. So see the point here. Some of the motors will detach, of course, and that's what we want to understand, right? Okay. So we need the attachment rate. You understand why we need the attachment rate? Because we want to end the run. We need the attachment rate of the opposing motor. To end the run, you need the attachment rate of the opposing motor. To end the tug of war, you need the detachment rate of the opposing motor. Because first you need the attachment rate, then you need the detachment rate, right? Then you need the force exerted per motor, right? Okay, because is this a force balance or not? Then you need the velocity, which I told you why you need it, because you want to go here and then is there something else you need? Right, I think that's all you need. If you need something more, we'll invent it. Okay, so then you write this. Isn't it obvious? Okay, so the jokes apart, this is a LabVIEW program. Don't worry about the voice. This is a pictorial way of writing a program. I'm sure that people like Gautam are smirking at this, but we write our programs in LabVIEW and it works for us. Okay, and you can write this in C, and we have done that also. But this program was written in LabVIEW. Don't worry about all these arrows and this and that. But here in this program, if you look at it, it looks like this. If you look at a C program, it will look like something else. But you need to give this program something. Okay, so you need to start somewhere, right? So what will you give it? Okay, let's start with Ambrish, I'm removing all your questions. Okay, what will we start with? When we start the simulation, what do we assume?
Initial conditions.
Okay, one person. You tell us.
Initial events, how many motors are there?
Right. So we have to start with some number of motors. Okay. So let us say we start with n number of dynein motors, and we start with n number of kinesin motors. Now this could be even be 0, one of them. Okay. So let us say with nk is equal to 0 we start. Okay. Now as
That's the figure. Nk is equal to 0 means no kinesin is attached to microtubule.
always, yes. Okay. Is this understood? Okay, we will not have repeats of this question then. If you ask this question, you have to come and do this. Okay, so this number of you could even have taken nd is equal to 0, depending on how you start. It's up to you. But the point is that when you do this kind of simulations over long times, irrespective of what your initial conditions are, you should end up with more or less the same, statistically the same results. Okay, that is important. Okay, then otherwise it doesn't make sense. Okay, so if I start with this and let's say I put ND is equal to 5. Okay? Will I be like that there? Right? I will move in the minus direction. Nothing is stopping me. And now I start with this and I now know that the probability of a kinesin attaching is some number. Okay? And what I do is that, exactly what Amrish was saying, I run my program and I assign probabilities to things and I check every delta t time, did a kinesin attach, did a kinesin attach, did a kinesin attach, maybe all the dyne is detached as well. That is also possible. Okay, so I have to check for all these things. Okay, so let us say that, so now the question is, how do I know the probability for kinesin attachment? Because once I start off here, that's what I need to know, right? I need to know the probability for kinesin attachment. Okay, how do I know that? That's slightly more difficult one, but let's try that. Okay, so to find that out, this is the best we could do. Maybe there are better ways of doing this, but this is the best we could do. What is this? This is this kind of an endosome stalling against an optical tract. You have seen this kind of data. Okay, so these are stalls. Goes out, pulls, comes back. Okay, so this is so this is either kinesin coated onto beads, single kinesin. Remember, okay, or kinesin on endosome, whatever you want, and this goes up and comes down. And now look at these square regions. Okay, there is. Let us measure this time from here to here, here to here, here to here, here to here. Okay, and plot the distribution. What does this tell you? Here, what has happened is that, so this is zero force, right? This, you people have seen optical trapping data. So this is zero position, which is at the center of the optical trap, which is when no force is being applied by any motor. Okay? Okay, so now, so what happened here was that the kinesin detached here. Clear? Okay, why am I doing that? The kinesin detached here. And it fell down here. Now it spent this much time before the kinesin reattached. Right? So I can get the attachment rate for kinesin from this. If I make the plot the histogram of these from here to here, if I plot a histogram of this, I will get something. Maybe I'll get an exponential function. Something. From that, I can plot the attachment rate for kinesin. I can calculate them. So if I calculate the attachment rate of kinesin, so let us say, what do we get from this? So this time from here to here is about 5 seconds, roughly. Okay. Let us say I get a mean value of 5 seconds. Then that tells me that kinesin takes 5 seconds to attach. Okay, so I can calculate a probability now. Okay, so if it attaches, you have to wait till 5 seconds before you attach again. Okay, it needs that much time. So that will tell you a probability per unit delta of reattachment.
So is there a comparison between the attachment rate of kinesin or dyne?
We'll come to that in a minute. Okay, we'll come to that in a minute. Okay.
The attachment rate will depend on number of motors present.
It will, but it will, yes, it will, because the other motors may hold. So there are 3 kinesin, the 3 dynes. They will hold each other in close proximity. So if a single one attaches, then it's possible it will just diffuse away. Okay, but so those are complications which we will not discuss right now. Okay, yes, that is completely true. So from this, do you understand that you can get an attachment rate? It takes 5 seconds to attach. Okay, so that this gives you an attachment rate, right? So just like that, you can do this kind of measurements for dyne also. Okay, and you can calculate the attachment rate. I've forgotten how much it is. So some number you will get. Now you need the detachment rate. Why do you need the detachment rate? Right. So to calculate how long this will last, right? Right. We need that detachment rate. How will I get the detachment rate from this data? Remember that this is detachment rate under load, isn't it? Because you're in a tug of war here. The kinesin is pulling you back. So Here's detachment rate under load. Okay, so I need the detachment rate under load for single dynein, multiple dyneins, whatever number of dyneins. How do I get that? From this data. Sorry?
So after the stall,
Now, tell me here.
reverse, yeah, when you're going back.
So this is, see, you are working against the trap. Think of the trap as kinesin. Okay, and you went up till here. If I measure this time, that is a measure of the detachment rate, right? If this time is long, goes till here, then the detachment rate is yes. So what you will see here is that you have a single dynein. This T stall is small, detachment rate is high, and if you have both, okay, this is single dynein because the force is less, 1 piconewton, two dyneins. So what you can do by measuring this, you can calculate the detachment rate for n dyneins under load. Approximately, you can always do better and better, but this is a good starting point. Okay, do you understand why we are able to calculate detachment rate for n dyneins? Because you know how much this went to. That tells you the force, and you know how much force single dynein exerts. You know this one is 4. So 4 dyneins survive for 4 seconds. Let us say 3 survive for 3, like that. So what we found is that actually 1 dynein survives for 0.2 seconds. However, 1 kinesin survives for 1 second, which makes sense, right? Because here you have 5 of them. So 0.2 each into 5, roughly 1, and things are not so simple always, but just a kind of, and here 1. So it is matched in what? Tenacity now. Tenacity under load. They are matched in force now, because this exerts 1 piconewton, that one is 5 piconewton. Okay, so that's one. Okay, so you can do the same thing for kinesin also. So you will get a detachment rate for kinesin. So what have we got till now? Absolutely from experimental data, no assumptions. Okay, you got a velocity for both motors, you got a detachment rate for both motors, you got an attachment rate for both. The attachment rate is a little dubious, and one has to think more about that. But remember that the attachment rate is assumed to be load independent, right? Because if you are in that vicinity, it doesn't matter how much load you are under, you will attach with your same rate. Now what happens after attachment is load dependent. But to go and attach, load doesn't matter if you are there, right?
In case of, we are considering that the kinesin is already attached to the load.
Kishanu, tell me when to stop. Okay. Yeah. Sorry. Yeah.
Well, you are slightly over time, but it's.
I'm already over time. Okay, I'll stop in 5 minutes. Yes, Pavel. Can we take the questions later, because I will never be able to finish. Okay, so now what we wanted to check was that we wanted to verify if by modeling, if this is the only situation, of course this is by modeling, this is the only situation which will give you this. What is this? This is one strong kinesin against multiple weak dyneins. And what is this? This is strongly biased minus motion with occasional tug of force. If we now see, we can't change the properties. Here is where building a model helps you. Okay, you can change the properties of the dynein and kinesin motors in a model. In real life, it is much more difficult to do that. Okay, you can change the properties of these and find out these trajectories and try to figure out if it will work or not. Right? So what did we do first? Is that so here what you see is that there is an asymmetry. Okay, what are the asymmetries? There is an asymmetry in numbers, 1 versus 5. There is an asymmetry in the single molecule function. This is much stronger. These are weaker. This stays on for long, but these stay on for a shorter time. But some of these can stay on for a longer time and max. Here there is a point which Ambarish was also mentioning about the catch bond behavior of dynein, which is another, I will talk about that. I have a talk in Mahabaleshwar.
Yes.
Yeah, then I will talk about that over there. About the catch bond behavior of about dynein. But basically, if you have more and more dyneins, then because of a catch bond like property, the tenacity keeps on increasing. That does not happen for kinesin. I think Amrish, you talked about that, right? Yes. So maybe we'll discuss this later. But anyway, so if you have a situation where now we make the dyneins like kinesins. Okay, what do? I make the dynein 5 piconewton force. I make the dynein detachment rate artificially 1 second. You know, it takes 1 second to detach, just like kinesin. And I put 2 dyneins and 2 kinesins on that. This will be the trajectory that you simulate. Now here, I think you cannot read the numbers, but this is in microns, okay? And this is in seconds. So it takes 500 seconds to move 20 microns, right? This is 500 here. I'll just read it out for you. It's 500 seconds to move 20 microns only. This is much lower than 1 micron per second velocity, 2 microns per second velocity. So it is not going anywhere. Okay, why is that so? They are constantly pulling against each other. Okay. Now in this simulation, we did not have the possibility that things can break apart together. We didn't implement that, but that's not relevant here. Okay. Now we can change other things. So at least from the modeling point of view, we can rule out the possibility that these two teams can be symmetric. Right? That is a symmetric situation. 2 dyneins versus 2 kinesins, both equally strong, everything same. Doesn't work. You go nowhere. Kind of expected, right? Now you change something. So here I had put, a kind of large, you can do this for a fairly large 2 dyneins and 2 kinesins. You can say, okay, let's reduce the number of motors. Both like kinesin, both motors, but I'll have very small number of motors there. Then you will have segments where the opposite one will not, once you attach one kind, it will take more time for the other one to attach, right? So you will keep going in one direction, but then you will reverse a lot. Here again the velocity is low, something like that. It's still low. It's still low, okay? So you can play around with this, and both like dynein low concentration, again, goes nowhere. Both like dynein high concentration. Okay, here what's the velocity? I don't remember what we got. Here also the velocity is low. 100 microns in 500 seconds. Okay. Now what we found is that when we used the actual parameters that we measured experimentally, is there anything unclear?
What is ANC?
Okay. Asymmetric motor competition model. Stock was not prepared for this. So asymmetric motor competition model is this. Asymmetric motors in competition. Okay. So when we put in these values which we measured experimentally, this one is weak, that one is blah blah, whatever I told you, then we get something like 4. So this is a simulated trajectory. Then we get exactly what we saw. Now this doesn't mean that this is what is happening. It's just that if you build a model like this, it agrees with the experimental data. It is not proof that that's what is going on. So this is something which I want all of you to really understand very well. Just because something fits to a model doesn't mean that is what is happening. There is a universe of other models which may fit to that. Okay. So this is what we saw, and then there were other things that we simulated, and then I think that should stop. And this we wanted to put in our paper, but we never did, is that you can build the entire 3-dimensional force velocity occupancy of the what is going to happen given any arbitrary number of kinesins and dyneins. You can plot this, and you can build up the what is it called? The phase diagram of this kind of motion. Okay, in 3 dimensions. These being the 3 dimensions. Okay, but I'll not do that. I'll stop here, Krishna.
Yeah.
So what I hope you understand or appreciate is that you can actually try and take experimental data and without much assumptions try and build. I mean, it doesn't always work. Here we were lucky it worked. Okay, you might not have such experimental data. There may be some caveats in using experimental data also, but if you want to play around with this, you can do it.
So, just for your information, your talk is the first one in Mahabaleshwar.
Okay, I'll talk on something else.
Okay, so we'll take a break now for 10 minutes. This is Pramod. So he will be your next speaker. We'll see you.